Good evening, this is Mr. Wintermelon. So in the last video, we have learned about the structure of our nephron. This will be important for us to understand what is ultrafiltration as well as selective reabsorption. And they are important for the formation of urine, which we are going to learn today. So by the end of this lesson, you should be able to outline the function of nephron with reference to ultrafiltration as well as selective reabsorption in the production of urine. So as what the objective have mentioned, there are two main processes which are involved in the formation of urine. They are ultrafiltration as well as selective reabsorption. So copy this down. Ultrafiltration is the removal of small molecules from our blood, and selective reabsorption is we selectively absorb back or reabsorb back useful substance. So the word selective itself means that the body is selecting what to be absorbed back. And this is largely thanks to the membrane channel that is present inside the site of selective reabsorption, which is our convoluted tubules as well as loop of Henle. And another important process that allows us to select what to absorb is active transport. So this process requires energy. The diagram on the right shows the glomerulus as well as the Bowman capsule, and this is the site of ultrafiltration. The force behind ultrafiltration is the high hydrostatic blood pressure. The high blood pressure is due to the contraction of our left ventricle. And if you look carefully at the two arterioles over there, you will see that there is a difference in the size of their diameter. So the arteriole that is supplying blood into the glomerulus actually have a wider lumen compared to the arteriole that is draining blood away from our glomerulus. And this will help to increase the blood pressure. Why is this the case? Imagine we have a hose down here. If we squeeze the end of the hose, you can see that the pressure will build up and water will squirt out instead of just leaking out from the leaky hose. So the difference in diameter will help to increase the blood pressure. Now, if you ask me, this is not exactly very important. It's good to know, but the important thing to understand down here is the pressure that drives ultrafiltration is due to the contraction of our heart muscle. And the high blood pressure will cause the filtrate to leave our blood. So what are the filtrate? So filtration will occur because our capillary is porous and the basal membrane of the Bowman capsule is partially permeable, as you can see from the diagram on the right. So what will be filtered out will be water, small solutes such as our salt, sugar, amino acid, as well as nitrogenous waste. Things that stays inside our blood will be red blood cell, white blood cells, because they are too big to pass through our blood capillary, as well as protein and fats, as well as other large molecules. So the important things to take note is what is coming out and what is staying inside. What is coming out is like your glucose as well as nitrogenous waste, specifically urea. What is staying inside is your red blood cell as well as your protein, because they are too big. So after being filtered out from our glomerulus, the filtrate will enter our proximal convoluted tubule. So in proximal convoluted tubule, selective reabsorption will take place. The word selective means that only specific substance is absorbed back. So water will be absorbed back by osmosis. Mineral salt as well as our nutrients such as glucose and amino acid, because they are essential, they are important to us, they are also reabsorbed back either by diffusion or by active transport. So these are the things that are absorbed back. Please copy this down. Then in loop of Henle and distal convoluted tubule, reabsorption will also take place. And they are by osmosis or active transport. As you can see, I did not highlight those because it is the same as proximal convoluted tubule. The only difference is further down, because the concentration of the glucose and amino acid, they become more concentrated. Since the water is reabsorbed back, they have to be recovered through active transport. Okay, lastly is the important part, which is the collecting duct. This is the site where ADH, remember ADH, antidiuretic hormone, have its effect. So inside our collecting duct, some water will be reabsorbed back, and the amount of water which collecting duct reabsorbed back is affected by our ADH. So the remaining water, as well as waste such as urea, will be passed out from our collecting duct. So this is the process of selective reabsorption. Things that we want, we absorb back. Things that we do not want, such as our urea, will be passed out from our collecting duct. So just to summarize the sequence of events, high pressure blood will come in from the renal arteriole. This will result in ultrafiltration of the glomerulus blood into the Bowman capsule. Now, it's not all of the blood is being filtered out. Only small molecules will pass through the capillary as well as the basal membrane and get filtered out into the Bowman capsule. And of course, water will be filtered out as well. So the filtrate will pass through the convoluted tubule and the loop of Henle. And selective reabsorption will take place. So those useful substances will be reabsorbed back into the blood capillary through diffusion, active transport, and of course, water will be through osmosis. And excess water and waste will flow out from the collecting duct. And to show you the animation, imagine this is glucose molecules. So what will happen is it will get squeezed through the glomerulus into the Bowman capsule during ultrafiltration, and it will flow through the convoluted tubule. So water as well as some nutrient would have been already absorbed, while the rest will sooner or later be absorbed in the loop of Henle or distal convoluted tubule. They will be reabsorbed back into the blood capillary and get transported out from the venule. In the case of nitrogenous waste such as our urea, what will happen is also the same. They will get filtered out during ultrafiltration, but the difference is instead of getting reabsorbed back, they will flow out through the collecting duct. And at the same time, large molecules such as our protein will not be filtered out through ultrafiltration. They will pass through the capillary and join back with the reabsorbed substances in the vein. And this is more of just for your information of things that are inside our urine, you can see that urine is mostly made up of water. Around 95% or 96% is water. And inside your urine, there should not be any protein, because protein are too big to be filtered out. Please take note. Also, if you are healthy, you should not have glucose. Unfortunately, for diabetic patient, inside their urine, it contains glucose, which is why it attracts ants. The concentration of urea as well as other nitrogenous waste, if you compare to the plasma as well as filtrate, will be a lot higher. Why are they a lot higher than the filtrate? Is because water is being reabsorbed back, and the urine is a more concentrated form of the filtrate. Other things that are found inside the urine are your salt, sodium chloride, as well as potassium. So this will be all for the two important processes that goes into the formation of urine. Ultrafiltration, powered by the high blood pressure caused by the contraction of our heart muscle, as well as selective reabsorption, whereby our convoluted tubules as well as loop of Henle reabsorb back nutrients and water, while leaving the waste such as urea inside the filtrate.